3 needle pines are really common in the landscape. They're an important tree for ornamental horticulture. Oftentimes, though, 2 and 3 needle pines show branch dieback, especially ones that are 10 or 12 years old. This is a typical tree showing those sorts of symptoms, where the lower branches are starting to show some tip dieback, and some of the branches have already been removed because they were severely affected. At the same time, the upper part of the tree is in pretty good shape. Now, this is caused by a disease called Diplodia tip blight, or now it's more correctly called sphaeropsis tip blight. Diplodia is an old name for a fungus that is now called sphaeropsis. This is a fairly easy disease to diagnose. One nice part about it is that it is most severe on the lower part of the tree, so you can get a good look at a sample from it. And because it's low on the tree, that's a clue as to what disease, or that a disease might be occurring. When a fungus is involved with a tree disease, oftentimes it's on the lower part of the tree. There's 2 reasons for that. One is the fungus tends to be most severe on the lower part of the tree, where the fungus is surviving from year to year, and as it rains, the spores are washed down to the lower parts of the tree. So these lower branches are getting spore showers every year. The other thing is you tend to have higher humidity and longer periods of needle wetness on the low part of the tree, and so the disease tends to be most severe down in this area. To diagnose the disease, all you need to do is look at the dead tips or the dying tips. One thing that you'll notice on a tip such as we have here, these needles were killed before they reached full size. And also, the needle is completely brown and dead. Ozone, which is an air pollutant, can kill needles, but normally what ozone does is kill just the needle tip, and the base of the needle remains green. Whereas with sphaeropsis twig blight, the entire needle is killed. Also, the fungus will move into larger twigs and kill those, and eventually into even larger branches. Now, to further confirm that you've got diplodia, if you look at these needles that were killed before they reached full size, there are little black fungal fruiting structures where the fungus is forming new spores, just peppering the surface of that needle. They look like little black dots or very dark brown dots, and you can see them with the unaided eye, but if you have a magnifying glass, that's helpful too. Now, what happens is the fungus invades those new needles just as they're beginning to emerge in the springtime, and the fungus quickly grows into the twig and kills the twig just below the bud. Sometimes you'll see resin flowing from that twig, where the fungus has killed the twig, the bark is dying and drying out, and as it shrinks, it cracks open and resin flows out. The other part of the tree that becomes infected are the cones. Now, the cones also will have fungal fruiting structures on them. This one's a little too young to show it, but some of the others do have the fungal fruiting structures. But the problem with this fungus is that it stays on these cones, and it stays on the needles, and neither the cones nor the needles fall off readily, and so the fungus gradually builds up in the tree. Now, trees of all ages are susceptible, but what happens is as the tree ages and you get more and more of the fungus involved on the tree, then it's the 10 and 12-year-old trees that start showing really severe symptoms. Then it starts moving up the tree and you get a major branch dieback. Now, the trees that are most susceptible are the ones that are under drought stress. This is really a canker-causing disease. It gets in the twig, girdles the twig, and kills it, and trees under drought stress are weakened and most susceptible to canker-causing fungi. To control this particular disease, the first thing to do is prune out infected branches. Especially on a tree like this where there's not an awful lot of infected branches, you should be able to go through and cut that material out. Then the next thing to do is to start a spray program. Now, the thing to remember is the only time infection occurs is when the new needles are emerging from the bud. So the spray has to be on those needles as they emerge, which means the spray has to go on during bud break. Usually one spray at bud break and then a second spray about 10 days later should be sufficient. Once the needles reach full size, they should be resistant. Sometimes when we get spring weather where the needles start to come out and then it's cold for a while and the needles are not fully mature for a couple weeks, you might have to go on with a third spray to protect those needles, but normally 2 sprays should do it. The other thing is, late in the year, in the fall, when especially if you've had dry weather late in the summer, you should try to irrigate the trees so that they don't go into the winter under drought stress. And so between the spraying, the pruning, and the irrigation, you should be able to control diplodia, or it's now called sphaeropsis.